Hello everybody. Today we're gonna talk about, as I said in the first episode, the Krebs cycle and what happens in the ATP synthase in the mitochondria. Basically, we're talking about what mitochondria does. We're not gonna do a recap or something about the first episode, and now the glycolysis happened in a free space in the cell, and now the pyruvate that was made in there goes into the mitochondria. Let's say this is the mitochondria. Actually, let's draw it bigger. You know those tiny little molecule thingies? Tiny little long-looking molecule thingies with one hydrophobic and one hydrophobic end and one hydrophilic end. Those little molecules making up the cell's membranes and stuff. This also makes up the mitochondria's membrane, and so I'll draw it as I think it would take a long time to draw it, so let's just draw two lines here. And there's enzymes over here, which is basically the gate for the pyruvate and the other material for the ATP producing. And yeah, these are the gates, and here are a bunch of stuff. Well, there would be the pyruvates, yeah, I don't really fancy drawing them. Let's say this is a pyruvate, and well, there's gonna be a bit more than just one, I would think. There's two pyruvates, and there are two more oxygen molecules, and there are 4 electrons from the NADHs, and these are pumped through by the ATP, the thing that they're trying to produce. The ATP kicks these enzymes with the phosphate ends and makes these gateway enzyme thingies open up and makes these gets these things pumped through into the mitochondria. And it's not necessarily just two oxygen molecules. There needs to be a lot of them. These are oxygen. These two bulb thingies are oxygen molecules, and these get pumped through, and then what happens is the thing called Krebs cycle happens. This is very important, quite important. And before the Krebs cycle happens, I'll quickly draw a pyruvate molecule here, and this is a pyruvate, and now this gets turned into a thing called acetyl group. I don't know if this is how you pronounce it, but it is called acetyl group, and what that looks like is basically you pull off two oxygen atoms and one carbon atom, peel it off and turn it into CO2, and basically deposited as waste. I'm pretty sure that's what gets put into the red blood cells when you breathe out. The thing that comes out. I think that's the CO2 you produce in your body, and when you breathe out, that CO2 comes out because your body doesn't need them. And as you do that, some electrons, 2 electrons, are stored in the NAD plus, so it becomes NADH. NADH. One NADH gets produced in this when this happens, and this is what the acetyl group looks like. And then there's this here. The acetyl group looks like this. And now this gets put into the Krebs cycle. So I think I'm gonna draw it here. Actually, I'm not gonna draw it because there's a lot of things to draw, and so I'm just gonna explain it. And there's gonna be some pictures in the screen right now. There should be, and well, I mean not right now as I explain, but first, well, there's a acetyl group, and it goes into the first level of that cycle. The first molecule in there is called oxaloacetate, and then the acetyl group comes in and makes citrate, and then the citrate loses, turns a part of the molecule into CO2, and deposits it as waste, and while doing that, NAD plus rips off 2 electrons and stores it, and steals it or stores it, whatever, and then the molecule that's left again makes another CO2 molecule, and as doing that again makes another NADH, and then now the molecule has made 2 CO2 molecules and made it fly away. So now in the molecule there are 4 carbon molecules in the carbon atoms in there, and then the next stage is the molecule frees enough energy to stick a phosphate and ADP together. So extra ATP is made here, and then FAD is, as the book describes, a close cousin of NAD, and the FAD also stores high energy electrons. So now the FAD stores some energy from that molecule, and now into the molecule, extra water molecule gets combined and turns into another molecule, and now this molecule produces another NADH, and while doing that, turns back into oxaloacetate, and the cycle cycles again. It keeps on cycling, and as this keeps cycling, because here we pumped 2 pyruvates into the mitochondria. Now the cycle has turned twice. It has cycled twice, and now if you look at the cycle again, the whole thing has produced 10 NADHs and 2 FADHs. So actually it's FADH2. And what does that mean? The NADHs and FADH2s has stolen 24 electrons from the glucose, because the pyruvate was the acetyl group was firstly was turned into from pyruvate, and pyruvate was made from glucose. And as the book describes, 24 electrons is the max number of electrons you can steal from glucose. And yeah, so now you have now you stole the most energy you could have stolen from the glucose, and the other stuff are waste. And as we stole 24 electrons from the molecules, also 24 hydrogen ions also have been produced. So yeah, there's 24 protons that have been produced, and also there has been 6 CO2 molecules that have been produced. So now we go on to more mitochondria stuff, and I think I'm gonna draw here, if you can see it. Let's say this is the inner membrane of the mitochondria. Mitochondria has 2 layers of membranes. This is the first layer, and there's a more wrinkly layer inside. And that membrane is made of the same stuff as the other membranes. And now let's say here are all the stuff that has been produced, the NADHs and FADH2s, and now these come over to near the inner membrane, and on that membrane there are small little pumps. These pumps are electron powered, so the stuff has stolen a lot of electrons. Now there's a thing called electron transport chain, and that makes the electrons from all the things that's storing electrons flow through the pumps and then come out back to the oxygens. And so basically the electron is falling towards oxygen, and that is producing more energy because the the electron is going to the inner layer because the electron is getting pulled by the oxygen, and that is producing energy. I think I said that in the last episode. And as that happens, the electron flows, and that means the fact that the electrons have flowed, that means there was an electrical current going on over here. So that electrical current powers the pumps, and the pump pumps the 24 protons that are over here. How do I think that's 24. I could be wrong, but now these get pumped outside, and then there's a thing called ATP synthase. It's an enzyme, and it does ATP producing. A lot of ATP producing. Now, I'll draw it over here. And there's a very bulby end. And now, the protons are pumped out, but this part of the mitochondria is, I think it's full of protons. Maybe not full of protons, but a little bit positively charged. So now the hydrogen ions now want to get back. Now want to get back to where they were before, and this hole is open right now. So they all rush in towards the hole in the ATP synthase, and now these protons flow through the hole in the ATP synthase, and this flowing of the protons produces kinetic energy, which means the ATP synthase can steal that kinetic energy and use that energy to put on phosphates onto ADPs. So as the protons produce kinetic energy, these bulby part, the bulbous, free rooms to produce ATP. So, and maybe there's one, and there's one in the bag, and there's one, but whatever, the ATP now gets produced by the energy of the protons moving. So now 34 ATP gets produced. So that's a lot. I don't think I'm gonna draw that, but 34 is quite a lot. And now we think about how much ATP has been produced by one glucose molecule. So in the first stages of producing ATP, I said in the free space of the cell, some enzymes attack glucose, and if you count them, well, there are 3 first molecules attacking the glucose, and then there's a fourth enzyme that snaps the glucose in half. So that means 4 enzymes has worked. So basically 4 ATP has been used. So that means -4. And now we count all the ATP that has been produced and that has been used, and we add all them together. Well, firstly, when doing the glycolysis, when preparing for the glycolysis, there are enzymes that do stuff, and those enzymes put two phosphates on the glucose, and those two phosphates come from two ATPs. So two ATPs has been used up, and then while in glycolysis, two ATPs are produced. So that's plus 2, but as we saw here, two pyruvate molecules are used. So this glycolysis happens twice to both molecules, I think, both GAP and DHAP. And now, so the same thing happens twice. So 4 ATP. So the preparing minus 2 ATPs, and then plus 4 ATPs, and then, well, and then I said these go into the mitochondria, and that means the ATPs kick the pumping enzymes. So that would mean minus 2 ATPs, and then the Krebs cycle. As a Krebs cycle happens, one cycle means one extra ATP. So the cycle happened twice, 2 cycles. So 2 ATPs has been produced from the Krebs cycle. So and that means we're currently on. 2 ATP. Now, this ATP synthase produces with the hydrogen, and with the hydrogen moving with their kinetic energy, produces 34 ATP. So we add up the 2 and the 34, and what do you get? 36. 36 ATP. 36 whole ATP molecules has been produced from one single glucose molecule, and that's a lot. 36 ATP. Now, that would mean moving 36 enzymes. I think this is quite a lot. And now, and also, our body has a lot of cells. So the book says, in a second, our body produces 10 million ATP. And that's quite a lot, if you ask me. And that was the ATP producing of life, how life makes ATP, and how cells make ATP. And I think, oh, one thing more. These were all about eukaryotes, which means eukaryote cells has a nucleus and a bit of mitochondria, and also there are cells called prekaryotes. Prekaryotes have no nucleus and no mitochondria, but they do have an ATP synthase outside their cell membrane. So they do the hydrogen pumping thingies on the outsides of their cell, and that's how cells produce ATP to work. And also plants too. Plants do these stuff too, and like trees, for example, they do this all too. And the subject of this series is about trees, and for 2 episodes we talked about making ATP. So now the 3rd episode of the series is gonna be about trees, finally. Hooray! And I think I'm gonna end it here for the 2nd episode, because I think one subject has been explained through, and I think it's a good time to end it. And so I hope you enjoyed. And bye. Oh, and if you have any questions, you can leave it under the comments, and I will try my best to leave an answer to that comment. So, and do that if you want to. And again, hope you enjoyed, and bye.